Students, today we study some aspects of wood anatomy. In wood anatomy, we study ontogeny of secondary vascular tissues, growth rings, heartwood, types of heartwood, sapwood, and wood parenchyma. In most dicot and gymnosperms, the stems increase in girth by the activity of lateral meristem. A tree stem consists of three areas: pith, xylem, and bark. The central pith is usually bare, visible, and does not increase in size through the life of the tree. A cylinder of wood, known scientifically as xylem, rises in diameter with age and rate of growth. And finally, the bark sheath can be subdivided into inner bark and outer bark, two regions. New wood and inner bark are added each year by the activity of layer of dividing cells, which is sandwiched between the inner bark and sapwood. New bark production is relatively small compared with new wood production, and bark is continually being shed to outside of them. This in older trees, the greatest volume of the stem is wood. Since the new wood is added to the outside of existing wood, the oldest wood is close to the pith and the most recent is close to the bark. The ontogeny and origin process of secondary vascular tissue. The initiation of secondary vascular tissues occurs after the maturation of primary xylem and phloem. At this stage, the cambium is represented by a single layer of cells. The primary tissues lying outside the secondary vascular tissues are subjected to pressure from the inside by the enlarging secondary vascular cylinder. The primary phloem is crushed and become non-conducting. The cortex persists for a longer time as it increases in its circumference by cell division and cell expansion. The epidermis may also persist through the cell division and cell enlargement to accommodate the diameter increase as the result of secondary growth. But in most of the plants with continuous secondary growth, these outer tissues are completely crushed by the formation of perioderm. Within there during secondary growth, these dead peripheral rays are eventually eliminated by decay or by abscission. The secondary xylem in the stem of perennial plants commonly consists of concentric layers, each one of which represents a seasonal increment. In transverse section of the axis, these layers appear as rings and are called annual rings or growth rings. Each layer represents the growth of 1 year. The width of growth rings varies greatly and depends upon the rate of growth of tree. The diagram shown the in center dark concentric wood known as heartwood and outer layers known as sapwood. In regions with a pronounced cold season, the activity of cambium takes place only during the spring and summer season, thus giving rise to the growth in diameter of woody plants. In spring or summer, the cambium is more active and forms a greater number of vessels with wider cavities. In winter, at one season, however, there is a less need of vessels for shaft transport. The cambium is less active. Early wood. The wood developed in the summer or spring season is called spring wood or early wood. Early wood is inner layer of growth rings. The early wood zones of growth rings typically consists of thin walled, larger diameter cells and also appear lighter in color. Latwood, the wood formed in winter or autumn season, is known as autumn wood or latwood. Latwood is an outer layer of growth rings. Latwood joins thick-walled, smaller diameter cells and also appear darker or brown shade in color. Dendrochronology. This is a branch of science. Each annual ring corresponds to 1 year's growth. And on the basis of these rings, the age of a particular plant can easily be calculated. The determination of age of a tree by counting its annual ring is known as a dendrochronology. Sometimes two annual rings are formed in a single year, and in such cases, the counting of the annual rings does not show the correct age of the tree. This happens when perhaps because of the drought condition prevailed in the middle of a growing season. Tylosage. In many plants, the walls of the xylem vessels produce balloon-like outgrowth into the lumen of the vessels. These specific vessels with balloon-like outgrowths, known as tylosage. Usually these structures are formed in secondary xylem. Tylosage are formed by the enlargement of the pit membrane of the half-bordered pit present in between the parenchyma cell and a vessels or tracheids. Difference between hotwood and sapwood. Sapwood is the outer region of the old tree known as sapwood or albinum. The central region of the old tree is called hotwood and duramen. Sapwood consists of recently formed xylem element. It filled up with tannins, resins, gums and some other like substances. It is not hard and durable. It is hard and durable. This is of light color and contains some living cells also in the association of vessels and fibers. It looks black or dark brown due to the presence of various substances in it. Generally, the vessels are not plugged with tylosage. Usually, the vessels remain plugged with tylosage. This part of the stem performs the physiological activities such as conduction of water and nutrients, storage of food, etc. The function of hardwood is not of conduction. It gives only mechanical support to the them. Types of hardwood. Hardwood is also divided in many types. The first one we study ring porous hardwood. For some group of species like oaks and elms, the early wood late wood transition occurs abruptly and is very distinct. Within each growth ring, a band of large early wood vessels is clearly visible to the naked eye, after which a band of late wood vessels appears much smaller and requires the use of hand lens to see. The distribution of ring porous wood. Semi-ring porous hardwood. For another group of species like black walnut, butternut and hickory, the pore transition from large to small diameter within a growth ring is gradual. The pores in the early wood zone have a large diameter. They gradually decrease in size and pores enter the late wood zone. Diffuse porous hardwoods. The last group of species have vessels pores. They are uniform in size across the entire growth rings. These vessels are usually small, uniform in size and they are very difficult to see within the naked eye. Ring porous hardwood also divided in many following types: solitary pores, pore multiples, pore chains, nested pores, wavy bands. Sapwood, resin canals. The first step after making the determination that a wood species is a softwood due to the absence of pores is to inspect the cross-section surface for the presence of a resin canal. Resin canals are a tubular passage in wood that exude pitch or resin in seal of wounds that occur due to insects or mechanical damage. Resin canals most often occur near the latewood zone of the growth wings. Softwoods can be separated into two classifications based on the presence or absence of resin canals. Species that have resin canals include pines, spruces, larches, Douglas fir. The species that do not have resin canals include firs, hemlocks, cedars, redwood. Woods with resin canals are further separated into two groups: those with large resin canals, pines, and those with small resin canals, Douglas fir. Spruces and larch. Using a simple wood identification set to compare the size and number of resin canals of different species is useful in determining how much they can differ between species. Wood parenchyma. Parenchyma are small, thin-walled, longitudinal cells that provide food storage. These cells are sparse in softwoods but are often quite significant in hardwoods. Parenchyma are often very small and difficult to see. However, there are many species with visible and unique arrangements of parenchyma cells that offer a clear structural pictures for decisive identification. Two basic types of parenchyma, paratracheal and apotracheal. The major difference between them is that paratracheal parenchyma makes contact with the pores or vessel elements, while apotracheal parenchyma are separated from pores by fibers or rays. In most species, apotracheal parenchyma are not useful in identifying wood with just a hand lens. Paratracheal parenchyma appears in many forms and is often more useful for identification. The diagram shows the classification of parenchyma arrangements around hardwood pores. That's the some aspects of wood anatomy. Thanks, students.